In the previous session, we discussed about plasma proteins, the major plasma proteins, albumin, globulin, and fibrinogen. Now we are going to discuss about the functions of plasma proteins. There are several functions carried out by plasma proteins, and few of the important plasma protein functions are: first one, the maintenance of colloidal osmotic pressure. Plasma proteins, especially albumin, is responsible for the colloidal osmotic pressure of plasma, which is responsible for maintenance of plasma blood volume and body fluid distribution. And this important functions related to pressure is maintained by albumin. Transport function: several plasma proteins serve as carriers of various components in the blood, and few of the proteins functions are listed here. Albumin helps in the transport of bilirubin and free fatty acids, transfer in the substance iron, thyroxine binding prealbumin in the T3 and T4, retinol binding protein in retinol, thyroxine binding globulin in T3 and T4, T3 and T1, haptoglobin in hemoglobin, like that. Lipoproteins are helpful in triglyceride, phospholipid, and cholesterol transport. So, along with the transport function, some plasma proteins are having buffering action. Plasma proteins, mainly albumin, are the second most important buffer after bicarbonate in the blood, which maintains the pH and also pH homeostasis is maintained by buffering action. Nutritive value: plasma proteins, mainly albumin, serves as source of amino acid during limited amino acid supply. Whenever the protein intake or amino acid intake is less, in such a condition, the plasma albumin will provide its amino acids for the various body functions. Cells uptake albumin by pinocytosis and degrade it to supply amino acids for the required function. And plasma protein in that fibrinogen helps in the blood clotting. Fibrinogen participate in blood clotting. These are the fewer functions that plasma proteins are going to perform. Next, we are going to discuss about electrophoresis. What is electrophoresis? Why it is used? What is its importance? Let us discuss. Electro means electricity or current. Pores means to move in a gel. So electrophoresis means movement of particles in a gel in a electric field. So if you ask question, as we just discussed now, the plasma proteins, especially albumin, globulin, and fibrinogen, and also in globulin we are having several fractions. These proteins that we discussed are invisible, we cannot see. And in plasma proteins, if we are talking about these different types of proteins, you can ask question, how you know the plasma contains these type of proteins even though they are invisible, and how you are separated? So for this question of separation and identification and quantification is with the help of electrophoresis. So the electrophoresis is a technique used to separate the various charged molecules in an electric field. So let us discuss this technique. Electrophoresis is a technique for the separation of charged particles under the influence of electric field. So charged particles can be separated in presence of electric field. The term electrophoresis literally means that the migration of charged particles under the influence of electric field. Charged particles means they are usually proteins, nucleic acids, amino acids, etc., which exhibit charges. Principle: many important biomolecules like amino acids, proteins, nucleic acids, nucleotides have ionizable groups and can exist as cations, that is positively charged particles, or anions, that is negatively charged particles. If you come across cations, for example COO minus, and then anions, cations with a positive charge NH3 plus and negative charge CO minus, depending on the pH of the medium. Under the influence of electric field, these charged particles migrate either to cathode or anode depending on the nature of net charge they possess. So these listed biomolecules in the first paragraph, you can see biomolecules like amino acid, proteins, nucleic acids, they possess a charge and that charge is having a different number of positive and negative charge. Based on that, their net charge, they will move to the respective electrodes. Cations moves towards cathode, that is negative charged electrode, and anions moves towards anode, that is positively charged electrode. So cations which are negative positive charge, they move towards cathode, that is negative charge, because positive likes negative, and anions which are negative charged, they will move towards positively charged electrode in an electric field of electrophoresis. The rate of migration depends on net charge, size, shape. So the movement depends upon these characteristics: net charge, size, and shape. When a when an electric field is applied, these charged molecules are separated based on net charge, size, and shape, because shape will also play a very important role along with size and charge. Electric field is removed before the particles reaches the electrode. Thus, electrophoresis is incomplete form of electrolysis. The separated particles are then located by staining technique with suitable dye. If you take a sample and subject it in electrophoresis, the sample which contains mixture of various type of biomolecules, and during the electrophoresis technique, the sample that you have taken which contains A, B, C, D, E, like that, say several biomolecules, they will get separated in different bands during the electrophoresis technique, and hence by seeing the band after staining, you can find out the how many types of biomolecules were there, and then even you can see the quantity of them based on the size of the band. A supporting medium is required to carry out electrophoresis. It can be filter, paper, or a glass slide coated with agar. So different types of electrophoresis technique are available: gel electrophoresis, paper electrophoresis, etc. You can carry out the different types of electrophoresis depending upon the type of sample you are taking. Technique of agar gel electrophoresis. Here we are going to discuss about agar gel electrophoresis. A small amount of sample is applied on the supporting medium, that is paper or glass, near one origin. The sample which contains different types of biomolecules is applied. The slide is kept on electrophoretic chamber. You can see in the diagram. So in a chamber there is a paper kept or a slide kept, and it is connected to the buffer solution, which is in turn is connected to electrodes. So the slide is kept on electrophoretic chamber and connected to the buffer compartment by filler paper strips. The current is applied through electrodes dipped in the each buffer compartment. This causes separation of sample into fractions. Their position are visualized after staining with a suitable dye. The rate of migration of each component depends on net charge, shape, and size. So in the diagram, if you see, you can see one is anode and another one is cathode. In the bottom you can see mentioned in the right side, and next is buffer, and then the paper and slide is there, which is attached to the buffer with a filter paper strip. The electrodes, which is cathode and anode, is subjected to current or electricity. The charges are moved through the filter paper along the paper or slide containing sample, and thereby separating the sample according to their net charge, shape, and size. Next is application of electrophoresis. It is a technique for the separation and quantification of plasma or serum proteins. Whenever we talk about plasma proteins, I have told that the types of proteins and also the quantity present. So how these proteins are identified and how their quantity is estimated or quantified, that is because with the help of these techniques. In separation of serum proteins, a buffer of pH 8.6 is used, which is greater than the isolated pH of all serum proteins. Hence, all the serum proteins have negative charge in the pH. So these serum proteins migrate to anode under electric field. This results in separation of serum into five fractions, namely albumin, alpha 2 globulin, alpha 1 globulin, beta and gamma globulins. These bands of proteins are visualized by staining with the stains like amido black or coma C blue. So these are the chemical names of certain stains which are used for staining technique. The width and intensity of each band is the measurement of concentration of each fractions. So if you see this illustration where the plasma proteins is electrophoresed, and after this electrophoresis you can see the bands albumin, alpha 1, alpha 2, beta and gamma globulin. The albumin, which is having a bigger band, indicates it is in large quantity. As we discussed, it is 60% of the plasma protein, along with the different fractions at different locations, indicates different types of proteins present. So you can see the charge in the left side negative, and right side you can see positive charge. So the albumin is towards the positive charge and gamma globin is towards the negative charge. So their net charge, shape and size made these proteins to from one location they get separated and sedimented in the different locations based on their net charge and shape. This is one of the separation technique. And next we are going to discuss electroporosis of serum proteins is useful in diagnosis of some diseases, its application. So in nephrotic syndrome, albumin is decreased, and chronic liver disease, albumin level is decreased. In infective hepatitis, gamma globulin and reduction of alpha and alpha 2. Like this, so many diseases can be detected by electrophoresis technique. And you can see in the last, this technique is useful in the separation of lipoproteins. When we talk about lipoproteins, HDL, LDL, VLDL and chylomicrons, that all these lipoproteins were identified by electrophoresis technique. Next is chromatography. Like electrophoresis, this is also one of the technique used for the separation of biomolecules. So let us discuss what chromatography means. Chroma means color, graphy means to separate, chromatography means separation of colored particles. Chromatography is a simple technique used for separation of individual components of a mixture based on differential distribution of these components between two phases. Whenever a sample is subjected to a two phases, that is stationary and mobile phase, based on their differential distribution, the samples will get separated and they can be identified by using certain stain techniques which gives color, and that that can be identified. There are many different types of chromatography techniques which use various principles like adsorption principle, partition, gel filtration or ion exchange principle for separation of different types of substances. So different types of biomolecules are there: carbohydrate, proteins, vitamins, minerals, nucleic acids, etc. For different types of biomolecule separation, we can use different types of chromatography techniques based on the samples. Among these, paper chromatography is most common type. So in the different types of chromatography, the paper chromatography is most common type. So let us discuss in detail what a paper chromatography means. In paper chromatography, the principle of partition is employed to carry out separation of individual components of mixture is achieved by differential relative solubility of these components in two phases. The cellulose fibers of papers act as a supporting matrix for stationary phase. For example, if you come across stationary phase means that will not move, and mobile phase means which will move. So whenever you take a paper, on that paper if you put a sample, and then the sample along with the paper, if you pass a solution, that is mobile phase solution on it, if the sample is more affinity to the paper, then it will not move much and it will stick to the paper. Whereas if it is having less affinity and to the paper and more affinity for the mobile phase, it will move along with the mobile phase. So that will make the separation. For example, if you take A, B and C sample in a solution, and if you put that sample on a paper chromatography, if A is liking the paper more, then it will not move much. If B is having 50-50 attraction for paper and the mobile phase, then it will move for the certain distance And if C is having more attraction for the mobile phase rather than the paper, it will move much more longer interval, because it is having a affinity or liking for the mobile phase. So based on this, A, B, C, which was spotted at the same place, while the movement of the mobile phase on the paper, they will get separated at the different locations. So this A, B, C, which are the samples, they can be identified, which are invisible, they can be made visible by using staining technique. This is about chromatography. Sequencing. If a protein is there, they are made up of amino acids, so we will tell this protein is made up of so much of amino acid. This is the first amino acid, this is second amino acid, the third amino acid is this one. Like that we can tell the amino acid from first to the last of the protein. So you can ask question, how you know the first amino acid is this one, and second and third and subsequent amino acid, how you can able to tell the names when the proteins are invisible. You cannot see the proteins. In that invisible proteins, you are telling the first, second, third, like this, the amino acid. How is it possible? It is all possible with the help of sequencing. The sequencing of amino acid in protein means the determination of number and order of amino acid in a protein chain. So number of amino acids under the order. Order means first, second, third, fourth, which is the amino acid is present, like that. That is called as sequence. The significance, structure and function of proteins have a very close relationship. Biological activity of a protein is attributed to the higher structure of protein, that is tertiary and quaternary structure, that we have already discussed. Primary structure of protein is all about number and sequence of amino acid. It determines the higher level of protein structure. Once you know the primary structure, we can easily identify the secondary and tertiary structures. Therefore, biological activity of a protein is in turn dependent on primary structure. So the primary structure, which talks about the number and sequence of amino acid, we can able to identify these by this sequencing technique. So sequencing of amino acid in a protein chain is an essential component in the study of structure and functional relationship of proteins. So how many amino acids are there, and which is the first, second, subsequent amino acids, we can easily identify primary structure of a protein by this technique. Process sequencing of amino acids is mainly based on Edmond reaction. So Edmond reaction is one of the technique used for the sequencing of proteins. Thank you very much.